Assalamu alaikum ji. Can you people hear me?
Yes, sir.
Okay. Acha ji, we will discuss replication today. So let's see, replication is duplication of DNA or synthesis of DNA using DNA as a template. Okay, so if DNA is used as a template and another DNA molecule is synthesized, this is called replication. There is another terminology. They should not be confused with it. It is reverse transcription. Okay, reverse transcription. The reverse transcription is also synthesis of DNA using mRNA as a template. Got it? So if DNA is made from, if mRNA to DNA is made, this is reverse transcription. And if DNA is duplicated from DNA, so if this is one DNA molecule, this is one DNA molecule, and two DNA molecules are made from this one DNA molecule, then this process is known as replication. Okay, there have been three theories proposed to explain this replication. Okay, it is number one, semi-conservative replication. And this semi-conservative replication is in our syllabus. The other theories are not to be described, or the other two theories are not in our syllabus. But to understand what's difference in, to clearly understand this semi-conservative replication, I'll make a comparison in between the other two theories. The second theory is conservative replication. Okay, and the third one is disruptive replication. Okay, so what is this semi-conservative replication? Okay, so semi-conservative replication is, if this is, for example, a DNA molecule, this is a DNA molecule. During semi-conservative replication, these two DNA strands are separated, and a new DNA strand is synthesized with each old DNA strand. So in this case, you see half of the DNA is conserved. Each DNA molecule has one old strand and one new strand. This is an old strand, and this is a new strand. So this type of replication is known as semi-conservative replication. So half of the DNA is conserved, or each DNA molecule made has one old strand and other new strand. So this is semi-conservative replication. What is this conservative replication? Conservative replication is that if this is, for example, a DNA molecule, both these strands of DNA molecules are conserved, and other DNA molecule made has both new DNA strands. Okay, so this is conservative replication. See, these both old DNA strands or old DNA molecule is conserved. So there is no change in this old DNA molecule, and this both strands in this DNA molecules are new. So this is a conservative replication. The whole DNA molecule is conserved. There is no change in the whole DNA molecule. And in this, is half of the DNA molecule is conserved. So that is why it is semi-conservative. And what was the idea of disruptive replication? Disruptive replication was, if this is, for example, a DNA molecule, each DNA molecule made has fragments of, I'll make a change. So this is, for example, two DNA molecules made, and each DNA molecule has fragments of, fragments of this. So there are fragments of old and new strands. So each DNA molecule had fragments of old DNA and fragments of new DNA. Each DNA molecule formed has fragments of old and new DNA. Later on, it was proved that these two theories were wrong. This conservative and the disruptive replication theories were rejected, and it was proven that the DNA replicates by semi-conservative replication. Okay. So we'll discuss, though we do not have directly these experiments to prove this semi-conservative replication. However, on this concepts, there are many questions given every year in the past papers. So that concept need to be understood clearly. So let's see experiments. To prove DNA replicates by semi-conservative method, not the other two. Okay. So this experiment could be done with bacteria. Two scientists carried out this experiment. They took E. coli bacteria. This is an E. coli bacterium. And cultured E. coli bacteria in a source of nitrogen, which provided N15 instead of N14. Okay. You know, there are two isotopes of nitrogen. There are two isotopes. Two isotopes. I'm sure you know what does isotopes mean. Isotopes of nitrogen. N15 is a heavier isotope. And N14 is a lighter isotope. Okay. So when they cultured this bacterium in heavier isotope for many generations, this was an N15 provided to this, you know, a source of nutrient. All the new bacteria made or produced by binary fission had DNA molecule. DNA in bacteria is circular DNA, which has, you know, the two strands. But I'll represent it with two lines. So these two lines represent that every bacterium has double-stranded DNA molecule. Each contains heavy isotope. So every bacterium contains DNA strands with N15. Okay. Now what they did, they isolated DNA from these bacteria, isolated DNA from these bacteria, and centrifuged in a cesium chloride solution. I'm sure you know what does centrifugation mean. Centrifugation is a technique where the particles, molecules, or substances are separated on the basis of their density gradient or on the basis of their molecular mass. So what they did was they centrifuged it. You know, this is a centrifuge tube. This is a centrifuge tube. Centrifuge tube. They took this isolated DNA into it and they spun it in the cesium chloride solution in a machine at very high speed, more than 30,000 cycles per second. And what happens? Cesium chloride and the normal DNA has the same density, whereas this heavier isotope has the higher density than cesium chloride, so it's settled at the bottom. Okay. And when they observed this DNA was found at this Z location. It was found at the bottom of this centrifuge tube. As it was heavier DNA, both strands were heavier, so it was found at the bottom of this centrifuge tube. In the next step, what they did, they cultured this bacterium in N14 for one generation, just once. Okay. So, you know, if these bacteria are cultured or allowed to grow or divide in N14 for one generation, there would be two bacteria made from one bacterium. This is one bacterium and this is two bacterium. Okay, they took this, you know, bacteria and they cultured it. This was at the beginning, at the start, and it divided just once. One bacterium divided by binary fission into two bacteria. So see what will happen. They were assuming that if it is semi-conservative replication, the two strands, these are the two strands of DNA. Two strands, two lines represent two strands of DNA. So these two strands will be separated in these bacteria. Okay. And each DNA molecule obviously has two strands. So the new strand will be formed from this N14. Okay. As this bacterium was provided N14 instead of N15. Okay. So this was cultured for one generation, first generation, and one bacterium that divided and produced two bacteria. They isolated this DNA again, isolated DNA, and centrifuged it in the cesium chloride again. And what they observed, if it had been semi-conservative replication, where now the new band should have been formed. Okay. They observed that the new DNA band was not formed at this Z position. Instead, it was formed at Y position. Why there was no DNA at this Z position? And why there was a band at Y position? The reason was each DNA molecule had one strand heavier and one strand lighter. So as compared to this DNA molecule, which had both heavier strands, that was heavier than this hybrid DNA. So that is how it settled in the middle of this centrifuge tube. Okay. So this was hybrid DNA. If it had been conservative replication, this is when it was semi-conservative replication. Conservative replication. But if it had been dispersive replication, similarly, the bands, they were expecting that they would have been formed at Y, but it had been if conservative replication, where the band should have been formed. If it had been true, the band should have been formed. Why? Because in case of conservative replication, the two DNA, the DNA molecules formed should have been in such a way that one DNA molecule had both the old strands and one had both the new strands. And the band should have been formed in case of conservative replication, one at an X position and one at Z position if it had been conservative replication. So as the band were not formed at X and Z, so this experiment proved or rejected the idea of conservative replication. Okay, so this experiment proved that DNA does not replicate by conservative replication. But still it was possible that DNA band formed at Y may be due to both semi-conservative replication and the disruptive replication. So they did another experiment. They cultured these bacteria for another generation in cultured in N14 for second generation. Okay, so जो फर्स्ट जनरेशन में बैक्टीरिया बने उनको फिर उन्होंने कल्चर किया for another generation in N14, and they were expecting that from one bacterium the two bacteria would be made, and each bacterium would receive one old strand and one new strand. You see this bacterium here, was if cultured. It had one red and one green line showing that one N15 and the other N14. These were two old strands. These were both old strands. And the new strands formed would have been made from N14 because it was provided N14. So the new strand here should have been N14. And here the new strand made should have been N14. So this is what they expected if it is semi-conservative replication. This is semi-conservative replication. And if they expected it is not semi-conservative replication, it is disruptive replication, then these two bacteria would have here fragments of old and new strands. To prove it, they isolated DNA and centrifuged it again. Isolated DNA and centrifuged again. Now, what did they observe? If it had been semi-conservative replication, so what were their observation? Their observation was that one band was formed at Y and other band was formed at X. So one band is formed at an X, other was formed at Y, and obviously there was no band formed at Z. So see, this proved that the DNA replicates by semi-conservative replication. If it had been replicating by disruptive replication, the band should have been formed at Y only, not at an X. So the formation of this band at an X proved that DNA replicates by proved DNA replicates by semi-conservative, not by disruptive. So both these theories were rejected and semi-conservative theory of semi-conservative replication was accepted by them. Hope you have understood it. Now let's see questions on this topic. Let me see if we have questions on this topic. I don't have questions in this sheet. Let me find a question. Just give me a minute. Okay, so this is question here on this topic. Bacteria were grown for many generations in a medium containing heavy isotope of nitrogen, which is N15 here. They were then transferred to a medium containing a light isotope of nitrogen, which is, this N14. And they were given time to replicate DNA and divide once. So they were allowed to divide once. Okay. So the idea is, if there was a bacterium which had been cultured for many generations, and N15, it will have both DNA strands with N15 and 15. So this is N15 and this is N15. Now this was cultured once in N14. When it was cultured in N14, it will divide into two bacteria. And when DNA replicated, each DNA molecule will have one old strand. This is an old strand. This is an old strand, and one will be the new strand, which is, so this is N15. This is an N15, and this is N14, and this is N14. So every new DNA molecule formed at the end of the first generation, this is first generation, will have 50% radioactive DNA and first 50% normal DNA. Okay. And what they did then, their DNA was extracted, spun in a centrifuge, and observed using ultraviolet light. The DNA with N15 settled at lower depth than the DNA with N14. Obviously, N15 was heavier, so that would settle at lower position, and N14 will settle at the upper position, which shows the predicted results after one generation, the medium with the light isotope. So after one generation. Okay, so this is after one generation. So one generation ke baad, the whole DNA or whole, the DNA made in all bacteria, that would be hybrid DNA. So this is hybrid DNA. Okay, which will have one DNA molecule of N15 and other of N14, and that would be lighter than N15 and N15, and will be heavier than N14 and N14. Okay, so what should be the correct choice? Can anybody answer? Are you people there?
Yes, sir.
So what should be the answer? So what result, if it is centrifuged, what results you would expect? You see, if both these strands are heavier, N15 and 15, then the DNA should be here. If one strand is heavier and other is lighter, it should be here. And when both strands are lighter, they should be N14, and N14 should be here. And this is an N14 and N15. So after first generation, after one generation, which DNA will be made, and how these bands would appear in this centrifuge tube. So you see, every DNA molecule will have one N15 and one N14. So this is where the DNA has one N14 and one N15. So this should be a correct choice. C should be the correct choice. Is it okay?
Yes, sir.
Now let's see, we have another question. See, this is another question. A culture of bacteria had all its DNA labeled with heavy isotopes of nitrogen. At the beginning, at the start, all bacteria had N15. So every bacterium had N15. So this is, for example, at the start. What they did then, the culture was then allowed to reproduce using nucleotides containing normal N14. So this was now cultured in N14. And from one bacterium, two bacteria will be made. This is one bacterium and this is another bacterium. So what will happen? Each bacterium will have one old strand and one new strand made of N14. The DNA was extracted, examined using a centrifuge after one generation. So this is after one generation. And again after two generations. फिर two generation में. So this is first generation, and this is second generation. Okay. In second generation, each bacterium will divide into two bacteria. So each will receive one old strand and one this other strand. And the new strand made will be from N14 because this was cultured in N14. So this is at the end of second generation and this is at the end of first generation. The diagram shows the position of DNA band at Z in the centrifuge tube when the DNA was first labeled. ये जो first label है, this is, the first bacterium. It showed this, it is heavier DNA, N15 and N15, so that would be here, N15 and N15. In which pattern would the DNA be found after the first and after the second generation? First generation में कहाँ पे होगा DNA और second generation में DNA कहाँ पे होगा? First generation में ये वाला DNA होगा और second generation में ये दो bands होंगे. Tell me what is the correct choice. After first generation, all DNA made will have one heavy and one lighter strand. This is one DNA molecule. And where would it settle? Because it is neither two strands lighter and nor the two strands heavier. So it will neither settle at Z nor it will settle at X. So where would it settle? It would settle at Y. And after second generation, one DNA molecule has both lighter strands. So lighter strand will settle at X. And one has hybrid. So that would settle at Y. So this is the correct choice. So C is the correct choice. Hope you have understood it. So that's it for today. We will continue with it with the process of replication in the next class, inshallah.
Sir, can you please share the notes in the group?
Okay, I'll share that. Okay, so I'm ending it now.